Hello, and welcome to the Access Journal webinar series. I'm Ann Edall with the Journal of Environmental Quality, and thank you for joining us today. Before we begin, I'd like to mention that you can enter your questions in the Q&A section, and the moderator will address them at the end of the presentation. The webinar will be recorded, and the recording will be sent to all attendees after the session. Now I'm happy to introduce our moderator for today's webinar, Dr. Tyler Groh, an associate editor with the Journal of Environmental Quality. Dr. Groh is a faculty member in the Ecosystem Science and Management Department at Penn State University. He conducts research focused on agricultural water quality, agricultural conservation practices, and stream health. Both Dr. Groh's research and extension programming seek to create solutions to real-world water quality issues through applicable science and hands-on experience. Welcome, Dr. Groh.
Thank you so much. And hello and welcome to the Access Journal webinar series. I'm your moderator, Tyler Groh. Thank you so much for taking your time to join us today. We are excited to continue this webinar series, which features the latest research published in ASA, CSSA, and SSA journals. Reminder that questions can be entered in the Q&A section, and we will address them at the very end of the presentation. Without further ado, I'd like to introduce today's speaker, Dr. Heather Preisendanz. Dr. Preisendanz received her BS in Civil and Environmental Engineering from Rutgers University and her MS and PhD in Civil and Environmental Engineering from Purdue. She joined the faculty in the Department of Agricultural and Biological Engineering at Penn State University in 2013, where she is currently an associate professor of natural resource engineering. Her research focuses on the occurrence, fate, transport, and potential impacts of contaminants of emerging concern in the environment. Today, Dr. Preisendanz will speak about the surveillance of wastewater treatment facilities during the COVID-19 pandemic. Many facilities began to monitor trends of SARS-CoV-2 in the wastewater as a way to understand the prevalence of COVID-19 in the community. Dr. Preisendanz and her team expanded surveillance efforts to include related pharmaceuticals, and they leveraged hospital data from the service area to better interpret the observed trends. The report was published in the September 2022 issue of Journal of Environmental Quality. Dr. Preisendanz, welcome, and the floor is yours.
Thank you so much, Tyler. I'm assuming you can see my slides and hear me okay, is that right? All right, awesome, sounds great. Thanks so much for joining us this morning. It's a real treat to be able to share this research with all of you. As you can see, we had quite a large range of people who worked on this project, so I'm happy to be able to present the results of the project today on behalf of all of them with you. And so if we could just transport ourselves back to where we were exactly 3 years ago in 2020. It's not a great memory, I know, but I think it sets the stage well for what we're going to be talking through today. And so if we look back at the headlines, wastewater surveillance was actually just starting to make the news in May of 2020. So you'll see a couple of examples of what the news headlines look like as wastewater surveillance efforts in the US and in other places around the world really started to pick up as an early warning system for the spread of COVID-19. And so this one article is from May 1st, 2020, and then this one's exactly 3 years ago, May 11th of 2020. And so it made me think I should go back and look at my calendar. What exactly was I doing 3 years ago today? And it turns out we had our very first Zoom call with the epidemiologists who began to start this project with us. So it was just in its infant stages exactly 3 years ago. And I must say that I let the team know that we were having the seminar today on the 3-year anniversary of when we started, and they were quite excited. So I thought that was just really something fun to share with all of you. And shortly after that, and I guess around August, is when the headlines started to break around how this data was being used for decision making. And so it was right around the beginning of the fall semester, as universities were starting to look at how to reopen safely and bring students back to campus, that this data really started to be used for more than just sort of the spread within a community, but decision making about what to do with students. And so I think just sort of remembering that perspective of where we were, you know, these 3 years ago is helpful for understanding where we are today. And so I'm not sure how many of you get to work with wastewater on a daily basis, or if this is just a fun seminar for you to tune into for today. But if you're familiar with wastewater, you know that there's all sorts of pathogens, nutrients, and chemicals that are in wastewater that are coming from domestic, business, industrial, hospital, and medical sources. And so these are all things that are coming to the treatment plant. And typically, what my research group does, and maybe some of you as well, is we monitor the Influent data, but really we only monitor the Influent data to understand what the effluent data should be compared to. And so that allows us to sort of look at the persistence of any of these chemicals or pathogens, nutrients, whatever you'd like to look for through the treatment plant. So we would bring those samples back to the lab, analyze them for the constituents of interest, and then think about what it means for those chemicals to be discharged to the environment. So this sort of down the pipe approach. So thinking about ecological risks of beneficial reuse of that wastewater for irrigation purposes or for other reuse activities. And then in other cases, directly discharging that treated effluent to a receiving water body. So to perhaps a stream or other surface water body or groundwater injection even of that treated wastewater. But what wastewater surveillance really allows us to do, in addition to doing those things on the effluent side, is sort of look up the pipe to understand what's coming in the influent to the treatment plant as an indicator of what the human health is in the community. So thinking about what that influent data means is more than just percent removal from the effluent perspective, but what it could tell us about the indications of human health within the service area. And so that was really the fun part of this project for us, was really starting to think about thinking about how to interpret the influent data differently. And so our study objectives were to investigate the relationships between SARS-CoV-2, which is the novel virus that causes COVID-19, and related pharmaceuticals of interest, to identify relationships between hospitalization data and constituents of interest, determine the effectiveness of wastewater treatment for removing pharmaceuticals from the influence, so this sort of traditional way of thinking about the data, and again, to understand the potential ecological risks posed by pharmaceuticals that remained in the effluent. You can imagine that because this is a human health emergency that we were going through at the time and that we're still not quite through completely, there was concern around the increased use of pharmaceuticals. We know treatment plants don't always remove those pharmaceuticals well, and so there could be some ecological risks that would be increased as the potential for as the usage of pharmaceuticals within our human population went up as a result of the pandemic. So there were some questions around what that might mean from a risk perspective for aquatic ecosystems. And so let me just talk to you a little bit about our water reclamation facilities that were our study sites. So we're located at the University Park campus for Penn State, which is sort of its main or largest campus. All of the treated wastewater from the university comes to this facility. It's actually been there in some capacity since 1930 or 1913, so way before most municipalities were required to have a treatment plant. We actually had one at University Park treating the university's wastewater. And then the surrounding area called the University Area Joint Authority Water Reclamation Facility. And so that treats this basically this donut-shaped service area. And so the whole is University Park, so that's Penn State service area, and then this area around it is what's treated at this University Area Joint Authority. And within UAJA's service area is a hospital, so Mount Nittany Medical Center. It's sort of our center county hospital, so 260 beds, 25 ventilator capacity, which was really critical at the time, crisis mode during the pandemic. Its capacity for treating wastewater is around 6 million gallons a day. And then for Penn State, so it's just actually getting the wastewater from only the University Park campus. So it's just treating Penn State's wastewater. And then its capacity is around 3 million gallons per day. So our overall approach was to take influent and effluent samples in collaboration with the operators at each of these facilities once a week. I should note, just as a side note, we did actually collect influent samples daily from both of these facilities over the course of probably the first year and a half or so of the pandemic. We did reduce that to 3 days after sort of things started to be a little bit more normal, at least on campus. And the data that I'm sharing with you today is just the weekly scale data, because we looked at pharmaceuticals only weekly. So I just wanted to note that in case you were wondering if we did this more frequently for virus analysis, weekly data wouldn't necessarily be sufficient to understand trends. So we did pull daily scale samples for just the virus alone. So each of those samples was then brought back to Penn State's labs. So we had to do some pasteurization on the influent samples because we were concerned about exposure of any microbes or pathogens, which obviously we were interested in looking at for this project, right? So we needed to make sure they were inactive before we were actually doing the analysis on the sample. So they did go through pasteurization just to make them safe for us to handle. We did have some increased safety protections in our lab at this time as well. We had some inspections come through just to make sure we were able to handle these samples appropriately. And then so we did extraction on the RNA and then qPCR analysis to look at the virus copies that were in the wastewater samples. And then the samples that we pulled for pharmaceuticals were subject through solid phase extraction cartridges, and then we looked at the concentrations of a variety of contaminants of interest that I'll talk about in a moment. But we actually didn't have, at the time of this project, the capacity to analyze the pharmaceuticals on campus. So we partnered with the USDA ARS location that's co-located at Penn State's University Park campus and worked with their analytical chemists and some of their hydrologists and water quality experts as well, who were collaborators on this project so that we could get that data. And so what we did with it was we shared it in as close to real time as we possibly could with the plant operations engineers, as well as other critical decision makers locally and on campus. And so we shared data probably within 24 to 36 hours of a sample collection for the virus, for the SARS-CoV-2 levels, so that data could be shared in enough time for it to be useful for decision making, if that was going to be something that the university would like to use it for, or the community. And then we also shared the pharmaceutical data as quickly as we could as well. The turnaround time for that was a little bit slower, probably 3 weeks or so between when we had the sample and when we could actually share the data back. It was hard to do lab work during this time. A lot of things were closed down. It was difficult to get equipment that we needed. And so just those kinds of challenges made it harder for us to share data back as quickly for the pharmaceuticals as we did for SARS-CoV-2. But still, that data wasn't seen as critical for decision making. So it was helpful for understanding what was going on in the community, but not as urgently needed with the operators as the SARS-CoV-2 data was. So looking at the target pharmaceutical analytes, we have been looking in my lab for quite a while now at different pharmaceuticals and personal care products. And so we just sort of started with the list of things that we were already sampling for. So at the time, that list included acetaminophen and naproxen, so the active ingredients in Tylenol and Aleve. And those were being used for if you had mild symptoms for SARS-CoV-2, that's typically what people were taking for reducing fevers and pain. So we looked at those constituents in the wastewater, and then we also added a suite of antibiotics. I know you're saying this is a virus infection. Why were we looking at antibiotics as something that is related to this project? We didn't expect this to have any relationship at all with the virus data, but we had already been searching or analyzing for these antibiotics already in wastewater, so we just continued to monitor for them. And also there was some discussion or some concerns that maybe people coming to the hospital could get secondary infections and then need antibiotic treatment. So there was some ambiguity about whether antibiotics usage would increase as a result of the pandemic or not. And then new for this project, we added dexamethasone, which is an anti-inflammatory that people who are on ventilators were often given. Remdesivir, which is the antiviral that was used to treat SARS-CoV-2 or COVID symptoms. And then hydroxychloroquine. I'll just sort of jump to the punchline on that one and let you know that we did not see it, but given the news that was breaking around hydroxychloroquine at the time, we just decided to add that to see if we actually ever saw it in our wastewater samples, and we did not ever see it. So I'll just start with the pain medication data that we had. So the series of graphs that you'll see in the next few slides, you'll see the dates across the bottom of each of the graphs. The pharmaceutical concentrations are shown in those blue bars, and then the concentrations are on the y-axis, the primary y-axis, and then the virus concentrations are the black dotted lines with the circles, and then those concentrations are shown in copies per milliliter on the secondary y-axis. And this was kind of a fun way to look at the over-the-counter pain medications. We actually just did some calculations to turn those concentrations into equivalent doses. So if you know the the wastewater treatment plant volume at the time, or the flow, then we could multiply the concentration in the flow and get a mass, and so then we could use that mass to convert it into doses. And so you can see in the Influent for Penn State's water reclamation facility, the number of doses of Tylenol that sort of increased through time. But this is pretty typical of seasonal usage of this of Tylenol going up sort of during the winter months. So we didn't see any correlation with virus concentrations, but it was still kind of interesting to see the equivalent doses over time for Tylenol leave. And it was also kind of interesting that they were around the same doses. We were trying to see if there was any indicator that there was more usage of one of these medications than the other, and we saw around the same doses, which I thought was just kind of funny that we were able to look at that. And then for the University Area Joint Authority Influent, so this, again, basically the same graph, acetaminophen on the top and naproxen on the bottom, and then the conversions of those concentrations into equivalent doses. And you'll see here, the numbers are quite a bit higher by about an order of magnitude. It is a larger service area, and it does have the hospital in its service area. So we can see that, those concentrations changing over time. So now I'll share the antibiotics data. So this is a little bit more condensed. So you're just looking at the 4 target antibiotics by row. So ampicillin, ofloxacin, sulfamethoxazole, and trimethoprim. And then Penn State's water reclamation facility data is on the left, and UAJA's data is on the right. And I'll note, just for convenience here, it's kind of useful to look at the data for Penn State through the lens of what was happening on campus. So you'll see the fall 2020 semester, students mostly had like a hybrid mode. So some students came back to campus in the fall. A lot of classes were taught remotely, so there weren't as many students on campus as we typically had. Then of course, winter break, where most students were home again. And then we actually had a delayed start to our in-person spring semester. We brought students back sort of in mid-February-ish. And so you can see the trends in when we saw antibiotics in the campus wastewater influent sort of broken down by semester schedule based on whether there were students here or not. And then, of course, that's not really true for the broader community, and we have a hospital again in our service area, so we have pretty consistent occurrences of each of those antibiotics across time, and again, really no correlations with the virus data. The next thing I'll talk about is Remdesivir. So you can see across the top, the Penn State Water Reclamation Facility data, the Influent, and then UAJA across the bottom. We plotted this with COVID cases instead of just concentration, so actually looking at case data in the service areas. And so again, it's not really clear that there's a correlation between new cases and the occurrence of Remdesivir. That's especially true on campus because we had cases, but we don't have a hospital in our service area, and so it was a little bit unclear where we're even getting Remdesivir and the Influent from. And so our team did a little bit of digging and asking around with our colleagues, and we actually found out that there was at least one researcher and potentially others who are using this experimentally in their labs. They were just doing some research with the chemical, and so of course it's going to the treatment plant where we're getting the effluent from everywhere on campus. And so that includes the labs and the wastewater from the labs. And of course, this isn't something that's regulated as a hazardous substance. So if it's being used at all in the lab, it's very likely that it's making its way to the treatment plant. So we thought that was kind of interesting. And the timing that we saw it corresponded to when they started their experiments, which was also kind of funny that we were able to sort of track down what the source was on campus. And then from UAJA, again, you can kind of see that there's not any clear trend between the new COVID cases and Remdesivir. But if we started to look through the lens of the hospitalization data, that was when things started to get a little bit more interesting. So if we looked at the number of hospitalized COVID-19 patients over time, we were starting to see some increasing concentrations, or at least occurrence of Remdesivir while the new cases were increasing. Our team did take a break during the winter months, or the Christmas holidays, winter holidays, just because we had been working literally every day since the summer to try to get this method and data off the ground. And so we just gave our team a bit of a break, and we did not capture samples during this time. So we probably likely missed some of the Remdesivir peaks that we would have seen if we had continued sampling. But again, I think this is just a nice way of saying, okay, well, we don't see any correlations with case numbers, but what if we look at the hospitalization data? Then do we start to see something? And that turned out to be the case for Remdesivir. It also was interesting for dexamethasone. So this is again at the UAJA facility. So we did not see any clear trends with dexamethasone concentrations in the virus concentration. But if we looked at the number of patients in the hospital who are on ventilators, not even just the number of COVID patients, but specifically those who are on ventilators, that's when the data again started to make a bit more sense. And we were able to say, okay, we're seeing dexamethasone peak around the times that there's increasing numbers of patients that are on ventilators. All right, so let me go back to the Influent and the Effluent data for comparison purposes. So how well does these two treatment plants remove these pharmaceuticals of interest? So you'll see acetaminophen and naproxen, the over-the-counter pain medications across the top. And these removal efficiencies, if it goes below 0, that just means that the effluent was actually higher than the influent concentrations. And so if it stays above 0, then that's the positive removal, so we're seeing lower effluent than the influent. And so you'll note that Tylenol and Aleve were removed well, so very good performance for removal of those over-the-counter pain medications, which I think in general is good news. You don't have to feel too bad then when you're needing to take a Tylenol. The treatment plant's able to remove that really well. And then for the antibiotics, we had quite mixed results, but that's just commonly true for antibiotics. Treatment plants don't typically tend to remove these well. So sometimes we were able to remove chemicals, other times not. Not really any clear reason as to why or any particular pattern that we had. So sort of mixed results in the data over time, with the lowest results for sulfamethoxazole, which is generally one of the harder compounds for treatment plants to remove. And then for remdesivir and dexamethasone, again, we did have some removal, more so at UAJA than from the Penn State facility, but both compounds did ultimately persist in the effluent. And so we were interested in potentially knowing what the effects of that might be. There's very little known about these compounds, especially remdesivir, since that's a newer pharmaceutical that we're used more because of the pandemic than it had been previously. Okay, so I just wanted to show you some of the concentrations that were in the effluent during the pandemic. So you can see just the range of concentrations for the over-the-counter medications in both of the facilities, and then for the pharmaceuticals as well as dexamethasone and remdesivir. And so obviously the lowest concentrations for those last two, but then higher concentrations for the antibiotics, and then highest for the or in general for the pain medications. All right, and so we need that data if we're going to think about the ecological risks. So we can use this data to look at these risk quotient calculations, and the common endpoints that are used are Daphnia, algae, and fish as indicators of the trophic levels of the food chain. And so this basically allows us to use the data that we're observing in the field, or in this case in the wastewater, and compare that to what would be considered a lethal dose or an effective concentrations where function would be inhibited. And we can do this for acute risk and also for chronic risk. And so this basically just allows us to say if there would be a short-term or long-term effect based on the concentrations that we observed. And so if you get a risk quotient that's greater than 1, then it's considered a high risk. A moderate risk would be a quotient between 0.1 and 1, and then a low risk would be between 0.01 and 0.1. And if you get a value that's below 0.01, then it's not considered to be a risk. So we looked at the data for acute toxicity and chronic toxicity for fish, Daphnia, and algae for the Penn State Water Reclamation Facility on the left and the University Joint Authority on the right. And what you'll see are the risk quotient values, and then the colors are just indicating the chemical that we were looking at. So the legends are down here for those colors. And you'll note that very few of the chemicals were present at highest at levels that were triggering a high risk. Most of them were either a low or a medium risk, and in many cases even just a low risk alone. So it just shows that the levels were not sufficiently high to typically cause a high risk to any of these ecological endpoints, at least for these two treatment facilities. But I mentioned earlier that at least the Penn State facility does beneficially reuse all of its treated wastewater for irrigation purposes. And so this allowed us to sort of assess what the ecosystem service benefits were of diverting that water away from Spring Creek, which is a high quality cold water fishery, and instead using it for land application instead. And so this just allowed us to talk about the benefits in a different way of this wastewater being reused and to mitigate those risks. All right, so I'll wrap up with some overall findings. So in general, pain medications and fever reducers were removed well by greater than 75% for both acetaminophen and naproxen. We had mixed results for the antibiotics, and we did see COVID-19 therapeutics in the effluent. So to some extent, the one treatment plant was able to remove them, and typically Penn State was not able to. The water reclamation facility there. And then the risk to aquatic organisms, sulfamethoxazole posed the highest acute risk, especially when students resumed normal activities in spring of 2021. That was when the sulfamethoxazole concentrations were the highest in the effluent. And then the living filter site does provide an important ecosystem service by beneficially reusing that treated wastewater. This is a picture from the facility. So we don't have a surface discharge permit for our treatment plant. All of it's irrigated, and so this allowed us to talk about those ecosystem service benefits. And then the risks posed by Remdesivir being in the effluent are really unknown. There's no data available on the concentrations that could cause adverse impacts, so there was no way for us to actually calculate a risk quotient for Remdesivir. And then I guess the last thing I'll note is that we can use the Influent as an indicator of human health. We were able to detect dexamethasone when it was co-occurring with increasing cases of patients with COVID-19 being on ventilators. And so I think this just sort of brought home that maybe just because you don't see correlations with the virus levels or even the number of new cases or even the number of people that are in the hospital with COVID-19, if you start to kind of figure out where these drugs are being used and then look at that data, then things started to make a bit more sense. So by looking at the number of patients on ventilators, we were really able to understand our dexamethasone data much better. So I'll just thank with the huge team of students, collaborators, and funders that we had for this project. We really had to do sort of all hands on deck, switch gears. The whole lab group sort of had to come together, form a team we had never worked with before with epidemiologists, leverage all of our existing partnerships. And it was only that really made this project a success. So I think that's it. That's it for the slides, and I'm happy to turn it back to Tyler to moderate the questions.
Excellent. Thank you so much, Heather. Great presentation. We do have quite a few questions here, so we'll get rolling with those. Did you ever, just for fun, test for ivermectin?
We didn't, because at the time when we were doing the study, that one hadn't hit the news yet. So we never added that to the suite of analytes we were looking at. But yeah, we did talk about what that might be interesting to do. I'd like to think that we wouldn't have seen it at least at Penn State's treatment plant, but I don't know whether we would have seen it in other treatment plants if we had looked for it. But it wasn't something we did.
Excellent. Thank you so much. This is a clarifying question. So were there no samples collected during winter break for Penn State's winter break?
Yeah, that's correct. So I'm trying to remember exactly which dates we stopped the collection, but right around the Christmas holiday, we just felt like the whole team just needed a break. We had really been going nonstop since May. I mean, I was putting in 14-hour days. I thought, oh, I'll be home with my daughter. I'll have so much more time to spend with her. And our whole team was cranking data nonstop. So I think even if we had samples during that time, I don't know if we would have analyzed them. But yeah, so we did not have samples during the break.
Got to have a healthy limit, right?
Yeah.
Is there any research being done on the solid side of things, like the solids from the wastewater plants?
Yeah, it's a great question. It's anything that doesn't wind up in the water effluent could wind up in the sludge. That's not something we looked at in the scope of this project. It just didn't naturally come out of the original goals that we had, which were really more focused on the influent side, and then tying it back to ag through the beneficial reuse. Clearly, there's a link back to ag and biosolids and land application. Yeah, it just wasn't something we were able to tackle during this part of the project. But maybe if this is useful information, Penn State's all of its biosolids are actually landfilled, so there is no land application of our biosolids. The copper levels are too high for us to be able to do that. So that's another story if we'd like to talk about that sometime. But yeah, and then all of the University Area Joint Authority composts their biosolids, and then that does get sold for use in the community. So it could be interesting to look at that.
Excellent. There's a question about, do you know what the difference is between the treatment plants and why the municipal one was able to remove more pharmaceuticals than the Penn State one was?
Yeah, actually, so that's a great question. The treatment plant was able to perform better at the University Area Joint Authority because they have activated carbon in their treatment train. And so they actually, because they divert, some of their water goes for beneficial reuse and some is directly discharged. So I was just making sure in my notes that I had the different treatment technologies correct. So the water that goes to beneficial reuse goes through microfiltration and then reverse osmosis. And anything that's directly discharged goes through activated carbon and then is disinfected with UV before it's just discharged. So those were the main differences. The treatment plant on campus has activated sludge and then chlorination. It doesn't have it. Well, at the time, I should just say we just brought in a new ultra membrane filtration system that came on board this February, which is super exciting for us, but it wasn't there yet at the time of this project. So.
Excellent. Thank you so much. Two more questions here. How were the data generated from this study used by the community partners?
Yeah, that's a great question. We had hoped that it would be used more than I think it ultimately was. I think our team was a little bit disappointed that there wasn't more enthusiasm around it. But I'll just say it's really noisy data. Trying to look at it, our team was like, do we see something? Do we not see something? Like the data was being shared with the provost's office on a daily basis. The team met with them and we shared the data. We were emailing the data to the treatment plant operators. I know it was discussed at emergency response teams locally. It just was really hard to tell what to do with it at the time. There's still not a good way to take the copies of SARS-CoV-2 that you see and turn that into the number of people that are infected, because the rates at which we excrete SARS-CoV-2 when there's an infection changes over the course of the infection. And so just doing that math is just challenging. So what we were able to do successfully, though, was when the university thought that they were starting to see dips in the numbers of testing cases that they were seeing on campus, there was a concern that students were evading the testing and going outside of Penn State's testing facilities to get their tests done. And so they were like, what are you guys seeing in the treatment plant? And we were able to see that the numbers were going down. so that was confirming to the university. So I would love to say that they did, that more was done with it, but I would say it was just really hard to in real time. But when it was most useful was confirming that there were in fact decreasing cases.
Excellent. All right, one more question for you, and we can wrap this one up. So now that society is more or less back to living normally, are wastewater surveillance efforts still ongoing?
Yeah, I think the value in doing this kind of surveillance has been really successful and really broadly recognized. And if for those of you who do surface water monitoring, you may have noticed that it's a lot more expensive to buy an auto sampler now than it used to be. It's because there's been such a demand by the treatment plants for an automated samplers to do this kind of surveillance efforts. And so I think because so much infrastructure and so much was invested in doing this initially, that there's an interest in keeping it ongoing for more than just SARS-CoV-2. So looking at flu, even looking at norovirus, and other, having the infrastructure set up to continue to monitor whatever the next new thing is of interest. So yes, I would say that this is definitely continuing, thankfully not at the daily scale that we were doing during the most intense portions. I think we're just doing 3 days a week now. It's just still a lot, but yeah, there's definitely interest in keeping these efforts ongoing.
Excellent. Well, thank you, and thank you everyone for joining us today for today's webinar. today's webinar, and to Dr. Preisendanz for sharing her research. Much appreciated. As a reminder, the recording will be made available and sent to all attendees very soon. Thanks again, and we hope you have a fantastic day. Goodbye, everybody.
Bye.